Good morning, everyone, and welcome to the webinar hosted by MedGenome. The topic for the presentation today is cell intrinsic and extrinsic factors for immunotherapy success. I would like to start by introducing our speakers today. We have two presenters. We have Dr. Lei Sheng, Associate Professor of Medicine from Johns Hopkins University School of Medicine. Dr. Lei Sheng is a physician-scientist at the Department of Oncology and Surgery at JHU and the Sidney Kimmel Cancer Center, and he also serves as the co-director of the Precision Medicine Center of Excellence for Pancreatic Cancer. His research focuses on understanding the complex biology of the tumor microenvironment by using pancreatic cancer as a research model, and he's involved in developing innovative therapies, including targeted as well as immunotherapy for cancer, pancreatic cancer, colorectal cancer, and other gastrointestinal malignancies. He's developed a number of mouse models of pancreatic cancer for dissecting mechanisms of metastasis and for preclinical developments of innovative immune-based therapies. And with that, I would like to pass along the presentation to Dr. Zhang.
Thank you, Akita. This is a really great opportunity for me to speak together with you. First, I want to give a overline of today's talk. So first, we're going to talk about opportunities in checkpoint inhibitor-based immunotherapy and the challenge with the current approaches, overview of the types of biomarkers in immunotherapy, tumor microenvironment as a biomarker for immunotherapy, methods to identify the biomarker and the limitation of the approaches, and then Ankita is going to give an overview of OncoPeptTUME, their bioinformatic platform. Then I will give a case study in pancreatic cancer, and then finally we're going to have a summary and a question and answer. So we are all in a very exciting moment. As you all know, FDA has improved many indications for pancreatic for immunocheckpoint inhibitor as a single agent in the last few years. I apologize. As you can see, each box will represent one indication of the checkpoint inhibitor. And I really couldn't find more space to put the most recent indications. However, as you can see in this slide, still the immune checkpoint inhibitor as single agents only approved for about 30% of all the malignant disease. There's still about 70% of the malignant disease types has no proved indication for the immune checkpoint inhibitor, particularly the anti-PD-1 or anti-PD-L1 antibodies. Overall, there's still only a small percentage of patients respond to the immune checkpoint inhibitor as a single agent. We estimate about 10 to 20% of patients. The majority of the patients, particularly solid tumor patients, 80% of patients still do not respond to the immune checkpoint inhibitor as a single agent. One of the reasons is we're still lack of a good predictor and a prognosis marker, as well as biomarker for the early response to the treatment. Now, saying we need to understand the mechanism of the sensitive and the resistant to the immune checkpoint inhibitor, so we can overcome the resistance to the immune checkpoint inhibitor as a single agent. If you look at the tumor microenvironments, just use the biomarkers that we know that can predict the response to immune checkpoint inhibitor in the best manner. For example, the staining of the anti-PD-L1 expression, or the tumor infiltrate lymphocyte, which is also correlated with the tumor mutation burden. And then we can see that the tumor microenvironment will tell us why there's only a fraction of patients that will respond to the immune checkpoint inhibitor as single agent. Those patients, about 20% of the solid tumor, their tumor microenvironment has a high expression of the PD-1 and also have a high level of infiltration of the tumor infiltrate lymphocyte. So those patients are likely to respond to the immune checkpoint inhibitor as a single agent. Majority of patients, their tumor microenvironment, lack of effector T cell, effector cells infiltration, and also is negative for PD-L1 expression. And those patients are unlikely to respond to the immune checkpoint inhibitor as a single agent, and also unlikely to respond to the current immuno-oncology combo strategies, because we still don't understand that this patient's tumor microenvironment very well. If we look at the tumors, they're both intrinsic factor and extrinsic factor that can influence the response to immunotherapy. For the best known, the intrinsic factor is something like a tumor mutation burden, the tumor expression of the PD-L1. For the extrinsic factor, for example, the components of the tumor microenvironment, many of those components are still we're poorly understand. One of the major reasons is the complexity of the tumor microenvironment. The tumor microenvironment is not a single cell. It's composed of the many different types of the cells, including the lymphocyte, T lymphocyte, B lymphocyte, and also myeloid cells. And other components were even much less studied. For example, the lymphatic vessel, the nerves, the fibroblast, and those components are particularly poorly understood, but they can serve as a barrier for the response to the immune checkpoint inhibitor as single agent. Therefore, it's very important to develop the technology, the method to comprehensively analyze the tumor microenvironment. Now I'm going to pass the speakerphone to Ankita, and she's going to give an overview of the current method to assess the tumor microenvironment.
Thank you, Dr. Zhang, for the overview of the current state of the immunotherapy and also the highlighting the importance of looking at the microenvironment. Of course, it's been in the current state of research, it is heavily studied, and these are some of the methods that are currently utilized. For example, there's immunohistochemistry that can be done on a multiplex, but there might be limited markers. There's also flow cytometry, for which you essentially need fresh tissue. Recently, single-cell RNA sequencing has emerged as a method of choice, but again, you need to have a lot of cells and they need to be viable. In a lot of the clinical cases, there isn't a lot of sample available, so hence, a method that can work well on fixed tissue or poor degraded tissue would be of choice. Now, RNA-seq for that would work, but sometimes if there is if there are genes expressed and shared across multiple cell types, it's difficult to identify the cell types. To solve this problem at MedGenome, we have built a pipeline called OncoPeptTUME, which uses minimal gene expression signatures of different immune cell types and of 8 different immune cell types to identify the cell types of interest. In the next few slides, I will review how the pipeline was built and how it was validated and show some case studies of how we applied OncoPeptTUME. So the way the OncoPeptTUME analysis works is you can start with RNA from freshly frozen RFFP tissues and do sequencing, and then do standard analysis to identify the genes of expression, and then I feed in the data for OncoPeptTUME analysis. And what this gives you is a cell type score or an immune score of all these different immune cell types that can be found in the tumor microenvironment. In addition to that, using the RNA-seq data, we can identify the pathways or the cytokines that are changing or altering the microenvironment. So how was the OncoPeptTUME built? We started by creating the gene signatures by utilizing gene expression data of microarray datasets of pure immune cell types. And then we applied a three-step approach to build the gene signatures. In the first step, we have ranked the genes by two different methods, the marker evaluation score and the average rank score. In the marker evaluation score, we determine the variability and plasticity of expression of every single gene that is present in these cell types across the different cell types, and we pick genes that are least plastic and less variable for our signature. In the average rank score, we rank the genes expressed in the samples based on high to low, and a gene that's expressed has a high rank would then be specific to that cell type of interest. After we did the filtering of these genes, we then correlated our gene list with publicly available RNA-seq data from the pure cell types, and the genes that were found to be highly correlated for each of the cell types were further reviewed using literature to confirm the function of these genes in the cell types of interest. A second criteria that was used to define the signatures for the gene of the cell types was to make sure that these genes are not expressed in any other cell type of interest represented in our pool, as well as it's not coming up upon exposure or any kind of stimulation. Once we had this signature, we then went on to validate the signature after we derived a minimal score or a rank score for the gene signatures. To validate our signatures, we have utilized data from 4 different platforms: RNA-seq data, FACS-based data, single-cell data, and additional microarray data from the immune cell types. Eventually, after all our analysis, we came up with a gene signature of a total of 42 genes for the 8 different cell types. So this is a table showing the different cell types that are represented and the data sets that were utilized to build the TUME pipeline. As you can see, there are cell types of the immune and adaptive immune system, innate and immune adaptive immune system. In the next 2 slides, I will discuss how we validated the signatures using RNA-seq and FACS datasets. So this plot here is showing an immune correlation of the immune score on the y-axis, and on the x-axis you can see the immune cell types and RNA-seq data. Each facet here represents a cell type, and what you can see is there is high correlation between the immune score and the cognate RNA-seq data of the cell type for every single cell type we have looked at, telling us that our gene signatures are highly specific for the cell types. Next, we tried to correlate our immune score with FACS-enriched datasets. And here, what you can see is the plot showing the correlation, where high is represented by blue and low correlation is represented in red. And the size of the circle represents the number of samples we have looked at. So what you see essentially is when we look at comparing the immune score versus the FACS-based signature, we see that there is a high correlation across all the different cell types except Treg. And an explanation for that is potentially the study that defines the Treg population might have used different markers than what we have in our signature. But in summary, using these 2 validation methods, we find that our signature is highly specific for the different immune cell types that can be found in the tumor microenvironment. We next tried to do a proof of concept study to identify or to evaluate the tumor microenvironment of 33 different cancers in samples found in TCGA data. So for that, we took 9,640 samples, and then we applied our OncoPeptTUME analysis and single sample gene set enrichment to derive immune scores for each of the cancer types. And these immune scores are now are sorted based on quartiles. And what each quartile represents is the relative enrichment of a cell type. For example, quartile 1 for a B cell would mean that there is high infiltration of B cell. The graph here on the right is an overview or a representation of the summary of the immune infiltration that we found for all these cancers. So on the x-axis are all the different cancer types, and on the y-axis are all the different immune cell types that we've looked at from this analysis. And what you can clearly see, for example, if you look at thymoma, in this case, if you look at the CD8 infiltration, there is majority of the samples here have a high CD8, which is what is expected. In another case for a diffuse cell B cell cancer, you actually see a high B cell infiltrate. Taken together from this analysis, what I'm demonstrating to you that one can look at the relative immune infiltration of all the different cell types at once and try and identify patterns of response versus, or build hypotheses to define mechanisms for response versus not. Now, to look further into the mechanisms of why certain patients would respond to checkpoint inhibitors versus not, what we did is we took all the data and we did a hierarchical clustering based on the immune scores that were derived from all the cancer types. And here we are going to focus further on cluster 4, which has a high CD8. Now, what we asked next is, in the patients that have high CD8, some have responded versus others have not. So what is it that is making them different? To look at that, we went back to our RNA-seq data and looked at the markers of expression, and then what we see is that between the alive versus the dead, there is CD8 infiltration in both, but in the live, that correlates with PD-1, but in the dead now, you have markers of exhaustion such as TIM3 LAG3, explaining that in the dead patients there is lack of activation. Next, we wanted to see what else is different in expression and signaling between the live versus dead samples. And what we found is that in the RNA from the live samples, there is upregulation of TCR signaling genes, indicating that there is active TCR signaling going on. We next utilized this data to look at co-infiltration of immune cells across different cancers, because it has been proposed in some studies that co-infiltration can also lead to survival benefit. So here what you're looking at is, so what we did is we took the top 20 and the bottom 20 of the tumors, of the responders versus none in the cancers, and then we did build a survival plot. On the top here is showing a case of kidney clear cell carcinoma, and on the bottom is low-grade glioma. What you can essentially see is, by looking at the infiltration of CD8 with CD4, etc., we see that there's a clear survival advantage when you have high CD4s in this case. Taken together from an analysis like this, we can define because we have the co-infiltration scores and we can look at it and identify markers of prognosis. Now I want to switch gears and move into a case study where we have taken samples of Lynch syndrome, or familial cases of Lynch syndrome, and applied OncoPeptTUME to study the microenvironment and then come up with a combination therapy regimen. So Lynch syndrome is a cancer where you develop a colon polyp, and upon mutations in the mismatch repair pathway, there is accelerated progression to colorectal carcinoma. Now, in this case, what we've done is we've taken some tumors and we have done OncoPeptTUME analysis to understand the immune infiltration. And what we can see for this particular patient here is that there is some CD8 infiltration, but there is a lot of MDSCs and macrophages of inhibitory immune cell types. So then we actually went ahead and applied our other pipeline for neoepitope vaccine prediction to find candidate neoepitopes, and we screened them using an assay to look at activation of CD8 T cells, and we see that some of the candidates like MH86 and PIGO are in fact immunogenic in our PBMC assay versus AXIN2 is tolerized. Now taken together, from this analysis and additional analysis for this particular type of case, we recommend a potential combination therapy with oxaliplatin and cisplatin. So now that I have demonstrated how OncoPeptTUME analysis can be utilized to define prognostic biomarkers as well as look at survival, I will pass the presentation back to Dr. Sheng to discuss his research.
Thank you. So Ankita just gave example of Lynch syndrome-associated cancer. So Lynch syndrome-associated cancer is a microsatellite unstable cancer. The hallmark is a high mutation burden as well as a high level of tumor-infiltrated lymphocyte. And their tumor microenvironment will look like what I show here in the right panel. This is done by multiplex immunohistochemistry. You can see there is a high level of infiltration of the CD8 T cells and higher level of the PD-L1 expression and a relatively low ratio of the FOXP3 T regular cell and the CD8. However, the immunohistochemistry are still lack of the method to quantify the MDSC and other myeloid cell at this moment. But the OncoPeptTUMEs, I'm very glad to see they are having the potential, the platform, to help us to analyze the MDC toward that direction. But the majority of the tumor type, as I said, that resist to the immune checkpoint inhibitor single agent, we call it cold tumor, and it shown at the left panel as it is represented to cold tumor. And you can see there's a fewer cells express the PD-L1 and a fewer effector T cell infiltration, such as CD8 cell, and a higher ratio of the FOXP3 positive T regular cell and effector T cells. And the tumor on the left panel is a pancreatic cancer. So, and then we collaborate with OncoPeptTUME and to analyze the tumor microenvironment of the pancreatic cancer. So, how we actually can, induce the T lymphocyte into the tumors? So we use a vaccine approach. This vaccine was developed in academic GMP by Lee Jaffe at Johns Hopkins. It's ready from on the shelf, and we show in the clinical trial had an excellent safe profile. And this vaccine is the whole cell vaccine composed of 2 allogeneic pancreatic cancer cell lines and express the GM-CSF. A few years ago, we did a clinical trial for the neoadjuvant treatment with this pancreatic cancer vaccine, GVAX. We give the patient who had a resected pancreatic cancer with the vaccine treatment 2 weeks prior to their surgical resection. And then we can take a look at their resected tumor and see how the vaccine can modulate tumor microenvironment. And we can see the vaccine can induce the infiltration of the lymphocyte and also form the organized lymphoid aggregate. This lymphoid aggregate has a tertiary lymphoid aggregate structure with organized T cell zone, for example, marked here by the CD3, and also the B cell zone that marked by the CD20s. Compared to the untreated pancreatic tumor, and there's only cluster lymphocyte, but not organized the structure of the lymphoid aggregates. Okay, so then we use a cohort of these patients. We also have their clinical outcome and the survival information. and we take their resected tumor, and we dissect the stroma tissue from the FFPE slice and subject them to the RNA sequencing analysis. But as you know, a mixed stroma specimen would not tell us the specific immune cell subtype. So we collected with the med genome and use their OncoPeptTUME platform and sort these cells in silicon into different immune subtype. As shown here is the lymphocyte panel. And Kita just already discussed how actually these cells are defined by the immune signature. And you can see majority of the tumors have a high level of CD4 T cell infiltration, but a low level of the CD8 T cell infiltration. And this is a panels of myeloid cell, including the monocyte and neutrophil, and NK cell, the macrophage, and also include the M1 macrophage and M2 macrophage. It will be best illustrated in the next slide to use the Z-score and display them in a heat map. And you can see the more blue colors represent a high infiltration, while the warm color here is represent the low infiltration of the immune cell. First, you can see the co-infiltration of the immune cells, and you also can see the trends of the immune cell infiltration in the pancreatic cancer tumor specimen. As you can tell, there's a high infiltration of the CD4, but a low infiltration of the CD8 cells, consistent with what we knowledge of the tumor microenvironment of a pancreatic cancer. And also, you can see the pancreatic tumor has a very low infiltration across all the patients with low infiltration of the NK cell, another effector cells, but has a relatively high level of the macrophage, particularly the M2 macrophage, but not M1 macrophage. So this is just at the beginning of the analysis. So what is the next step? And because the RNA sequencing also can give you the transcriptome of the whole genome, now you can correlate with each immune subtype as shown on the upper panel, as I just showed you before, and also correlate with each individual immune signature, cytokine profile, or as Ankita showed you the activation genes or the exhaustion genes. So then you can see how is the correlation between the immune subtype within the tumor microenvironment and how they correlate with the expression of these different cytokine profile or the immune gene signatures. So this is our example of using a cold tumor like pancreatic cancer and how we use this med genome platform to analyze the tumor microenvironment of pancreatic cancer. So in summary, the OncoPeptTUME RNA sequence-based platform that use highly specific minimal gene expression signature to predict relative enrichment of 8 immune cell subtypes in the tumors, include lymphocyte and myeloid cell subtypes. Now, OncoPeptTUME can be used for patient stratification, identifying prognosis markers, and guiding a combination therapy. And TCR signaling pathway genes, CTL, cytokines, T cell exhaustion status can also be used as a prognosis markers and for the to correlate with the T cell subtypes. And the immunohistochemistry profiling using the OncoPeptTUMEs of cancer can be utilized for guided combination therapy for the MSI high cancers, for example, Lynch syndrome associated cancer. And finally, the immunological profiling of the pancreatic cancer patients treated with the vaccine. We can use the OncoPeptTUME platform to show the co-infiltration of the high CD4 T cell but low CD8 T cell and the low NK cells, and the low M1 macrophage but a high M2 macrophage. Okay, now this is the summary of our talks. Now I'm going to pass the speaker to Ankita.
Thank you, Dr. Zhang, for a wonderful presentation, and I will end the webinar. We will move to the Q&A session, but before that, I just wanted to discuss that the OncoPep solutions at MedGenome, today we presented specifically OncoPeptTUME, which is looking at the biomarkers and the using the immune score. We also have an OncoPeptVac, which can be used to find therapeutic cancer vaccine candidates. Recently, as we all know, looking at the TCR repertoire and trying to understand how the TCR repertoire can respond to antigen-specific antigens in the tumor, and also in looking at the immune monitoring. So we are offering OncoPeptTCR as a service as well, and we also have a solution to look at the toxicity upon checkpoint inhibitor and adverse effects. Now, with that, I would like to thank everybody for attending and move to the Q&A session for this presentation. So the first question that we have has come in is, how does OncoPeptTUME differ from a nanostring pan-cancer? The OncoPeptTUME analysis has a relative enrichment uses RNA-seq data, so you can actually define the gene signatures, and we can also look at the broader expression and signaling of genes versus nanostring. The next question that has come in is for Dr. Zhang. So how do you plan to use your findings from this analysis for development of biomarkers for your research and for patient stratification? Dr. Zhang, if you could take that question.
So obviously what I just showed just the beginning of this project, and so our next step, as I showed, one of the next direction is this RNA sequencing will illustrate the transcriptome of the whole genome, and we can potentially, just as show some of the example Ankita has shown, we're going to look at within how attributes within each gene signature that define the immune cell subtype and of the specific patient, what is the level of the cytokine profile and also their, for example, T cell exhaustion and activation status, and to look at it based on that to see how they correlate with the patient's survival, and that will help us to define the a potentially new biomarker or treatment target. And other aspect will using this data to look at its relationship with other feature of what we just talked, the intrinsic factor and extrinsic factor, for example, the tumor mutation burden in the pancreatic cancer. We do have the tumor mutation burden status of this patient, and there's rare cases with a high mutation burden. We also want to understand because we didn't see in those cases with just a simple number of the change of the immune infiltration. As you can see, all the these patients seems to have a low CD8 T cell infiltration, but I want to see what they're actually in addition to the the CD8 T cell infiltration, whether it's also associated with other change of the expression of the immune signature. And I think that there's a many direction we can take. I just want to say this is a very good way to utilize the RNA sequencing data, so we can incorporate it together with other analysis that to look at the intrinsic and extrinsic factor of the tumor.
Okay, thank you, Dr. Zhang. There is another question that has come in. This is going to be the last question we'll take, and then we'll close the webinar. The question is, can OncoPeptTUME be used on RNA-seq from whole blood to deconvolute the immune cells? The answer to that is yes, we can do RNA-seq from the whole blood and try to, if you think there are immune cells in there that will correlate. If we have enough RNA, we can definitely utilize this approach for that. With that, I would like to close this webinar, and thank you all for joining, and we look forward to seeing you all next time. There are some questions that we haven't addressed, and they will be sent out. The answers to those will be sent out via email. And thank you, Dr. Zhang, for your presentation.
Thank you.